You're welcome to Excellent Simplified Tutorials.
Alright, so we've looked at the glycolytic pathway, and if you've not watched that video, please go down and watch the video. So this is a continuation of the glycolytic pathway. Now, the TCA cycle is also called the tricarboxylic acid cycle, or the Krebs cycle, or the citric acid cycle. Now, remember that we got pyruvate as the end product of glycolysis. Now, pyruvate was the end product. Now, this pyruvate will be converted to acetyl-CoA. That's the first reaction taking place here. Pyruvate will undergo what we call oxidative decarboxylation to acetyl-CoA. Now, this is what I mean. This is the structure of pyruvate. You can call it pyruvic acid. Now, if I remove this carboxylic acid group, this is called decarboxylation. Now, you agree with me that if this carboxylic acid is removed, what I will have left is just the acetyl group. So, to this acetyl group, coenzyme A is attached with a sulfide bond, and we get our acetyl-CoA. So, this is pyruvate and this is acetyl-CoA. So, from here, carbon dioxide is given off and coenzyme A is coming in to form our acetyl-CoA. This reaction actually is not part of the TCA cycle, but it's called the linked reaction between glycolysis and TCA cycle. Now, the acetyl-CoA in the mitochondria now combines with oxaloacetate. To form the first intermediate, which we call citrate. Now, the first intermediate in this pathway is citrate, and that is why the name of this pathway is called citric acid cycle. Citrate is the same thing as citric acid. That's why we call the name of the pathway citric acid cycle or tricarboxylic acid cycle, because citrate is a tricarboxylic acid. Now, the enzyme catalyzing this reaction is called citrate synthase. The name of the enzyme catalyzing this reaction is called citrate synthase. Now, from citrate can be converted to isocitrate. Citrate can be converted to isocitrate. And the name of the enzyme catalyzing this reaction is called aconitase. Aconitase. From citrate to isocitrate, the enzyme catalyzing that reaction is aconitase. Now, isocitrate will be converted to alpha ketoglutarate. Isocitrate will be converted to alpha ketoglutarate, and the name of the enzyme catalyzing this reaction is called isocitrate dehydrogenase. Isocitrate dehydrogenase. Now, there's something to note about this reaction here. In this reaction, NAD plus is converted to NADH. And this is very important because we use it. Now, alpha ketoglutarate is further converted to succinyl-CoA. Succinyl-CoA. And the name of the enzyme catalyzing this reaction is alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase is the enzyme catalyzing this reaction. Now, anytime you hear dehydrogenase, the enzyme dehydrogenase, that enzyme is involved in either oxidation or reduction reaction, redox reaction. So this enzyme, alpha ketoglutarate dehydrogenase, is involved in oxidation reaction here. And so in this particular reaction, NAD was also converted to NADH. Please take note of this because we are going to use it in our calculations. Here we produce one NADH. Here we also produce another molecule of NADH. Now, the next reaction taking place here is succinyl-CoA will be converted to succinate. Now, if you don't call it succinate, you can call it succinic acid. Just like if you don't call it pyruvate, you call it pyruvic acid. Now, from succinyl-CoA to succinate, NADH is not produced in this step. Yes, NADH is not produced in this step. And the name of the enzyme catalyzing this reaction is succinyl-CoA synthetase. Succinyl-CoA synthetase. Now, as you can observe, dehydrogenase is not part of the name of the enzyme, and so we are not producing NADH. This point, GTP is converted, GTP is produced in this step. Guanosine triphosphate is produced in this step. Now, from succinate, we started from pyruvate. Pyruvate was decarboxylated to acetyl-CoA. Acetyl-CoA condenses with oxaloacetate to form citrates. From citrate to isocitrate, isocitrate to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA, succinyl-CoA now to succinate. From succinate now, we have fumarate. Another important step here, fumarate. From succinate to fumarate, the name of the enzyme catalyzing this reaction is succinate dehydrogenase. And remember what I said about dehydrogenase. Succinate dehydrogenase. Remember what I said about dehydrogenase. It means either NADH is produced or FADH2 is produced. In this reaction here, what is produced is FADH2. This is very important because we are going to use it in our calculation. Now, from fumarate now to malate. From fumarate to malate, and the name of the reaction, the name of the enzyme here is fumarase. Nothing is produced here. And from malate back to oxaloacetate. The name of the enzyme catalyzing this reaction is malate dehydrogenase. We are seeing another dehydrogenase here, dehydrogenase, meaning NADH was produced. So here, NADH was produced. So if you look at this, this is just your tricarboxylic acid cycle. This all. Pyruvate to acetyl-CoA, which is the linked reaction. Acetyl-CoA condenses with oxaloacetate to form citrates, to isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, back to oxaloacetate. Now, I'll use a past question to analyze the energetics of this pathway. Alright, so let's look at this question. The question says, in the citric acid cycle, energy released during substrate level phosphorylation is catalyzed by dash. Is catalyzed by what enzyme? Now, in the glycolysis, we talked about substrate level phosphorylation and oxidative phosphorylation. Any step where dehydrogenase enzyme catalyzes, that reaction is actually oxidative phosphorylation. But if ATP or GTP is either produced or used, that reaction is substrate level phosphorylation. So in substrate level phosphorylation, we are either transferring a phosphate group from a substrate to GDP to form GTP, or we are transferring phosphate group from ADP. We are transferring phosphate group from the substrate to ADP to form ATP. Now this is what I mean. From the question, it says, which of them, which energy is, what enzyme catalyzes the reaction where substrate level phosphorylation releases energy? Now, in this step here, this is not substrate level phosphorylation because the name of the enzyme is dehydrogenase. This is oxidative phosphorylation. This is also oxidative phosphorylation. Now, the substrate level phosphorylation is seen where GTP was produced, guanosine triphosphate was produced. So the name of the enzyme catalyzing this reaction from succinyl-CoA to succinate is called succinyl-CoA synthetase. So the name of the enzyme catalyzing that reaction is called succinyl-CoA synthetase. So take note, if they say oxidative phosphorylation, then you can look at any of these pathway where NADH or FADH2 was produced. But if they say substrate level phosphorylation, check for GTP or ATP. That is a simple trick to getting it. So that's the name of the enzyme catalyzing that reaction.